Oxidative phosphorylation is the process in which cells use enzymes to oxidize nutrients, releasing chemical energy to produce adenosine triphosphate (ATP). This detailed explanation will cover its definition, steps, and regulation. 1. Definition and location. Oxidative phosphorylation (OXPHOS) is the final stage of cellular respiration in aerobic organisms. It occurs within the mitochondria of eukaryotic cells and the cell membrane of prokaryotes. This process converts the chemical energy stored in reduced electron carriers NADH and FOD2 into the readily usable energy of ATP. 2. Steps of oxidative phosphorylation. 1. Electron transport chain, etc. Electrons from NADH and FADH2, generated during glycolysis and the citric acid cycle, are passed along a series of protein complexes (IIV) embedded in the inner mitochondrial membrane. As electrons move through the etc., their energy is used to pump protons H+ from the mitochondrial matrix across the inner membrane to the intermembrane space, establishing a proton gradient. The final electron acceptor is oxygen O2, which is reduced to form water H2O. 2. Chemiosmosis. The proton gradient generated by the etc. creates a proton motive force PMF. This PMF is a combination of a chemical gradient difference in proton concentration and an electrical gradient difference in charge across the membrane. 3. ATP synthesis. Protons flow back down their concentration gradient across the inner mitochondrial membrane through a channel in ATP synthase complex V. The energy released by the proton flow drives the rotation of part of the enzyme and the synthesis of ATP from ADP and inorganic phosphate Pi. This process is known as chemiosmosis and is the mechanism by which the potential energy stored in the PMF is converted into chemical energy in the form of ATP. 3. Regulation of